Okay, so hi everyone again. I think we should probably get going. Welcome again. My name is Stoyan Stoichev. I'm part of Present Bio. My focus is on helping to develop some of our workflows in the proteomics space in particular. And today with me, I have the pleasure of hosting Dr. Magnus Jakobsson from the Department of Immunotechnology at Lund University. And so before we get going, just a couple of housekeeping announcements. We are recording this session, so we will make it available to everyone so they can view at their own leisure offline. Please post your questions using the chat Q&A option. We will have plenty of time to address these. And in fact, if you want, we can have your questions live to initiate a bit of a discussion after the presentation. So you can use the raise hand option, and then I'll try and activate or unmute your mic at the end of the presentation. And so before I introduce Magnus and his presentation, I want to give you a brief background and a bit of our thinking in terms of how this Emerge webinar series idea came about, and what would you like to we would like to achieve, and how we would like to build it over time. And I think in order to understand that, just a very brief background of what we're about, and hopefully to be able to highlight the importance of the contributions of the scientific community towards our offerings. So as a company, our main focus is on improving efficiency of sample prep, in particular in the areas of omics, mass spectrometry based proteomics, as well as bioseparation. And we are essentially sitting in a space between the samples and analytical, and dealing with the well-documented complexities or difficulties in terms of different sample sources, high complexity, wide dynamic range, low abundance, as well as poor ionization efficiency. And really our thinking about improving these workflows is quite simple. At the base, we want to improve the efficiency, to streamline these workflows. And we do this in different ways by integrating different types of chemistries, or in a more, what seems maybe a more trivial thing is to remove steps that could be bottlenecks, such as centrifugation, drying, buffer exchange, and so on. So these seem quite trivial, but once you start scaling up in terms of number of samples, these type of steps can be quite big bottlenecks and introduce quite a lot of variability into your workflow. So ultimately, our aim is to transition from manual, so from few samples to many samples, to be able to scale the process seamlessly, as well as to be able to transition from manual to automated workflows in a manner that doesn't require major reoptimization of the workflows. And so all of this must be underpinned by our thinking that all the workflows need to have the principles of robustness, high throughput, being able to be automated, as well as being able to be open. And what I mean by this is that we don't want to have any hidden buffer components, so everybody's understanding what the mechanisms of actions are, and you can then optimize your workflows where and if needed further. And so to highlight how we play a role in the space, and more importantly, what is the contribution of the omics community, our clients and collaborators, towards our offerings, and how this then plays back to this idea of the Emerge webinar series. I thought it's best to give you a highlight reel with some of the publications that we've been involved in the past. And so in the beginning, when we started developing the technology, we weren't really thinking about proteomics so much. Our focus was mostly about bioseparation and biocatalysis. But even then, the thinking about high throughput, efficiency, ability to automate was always central. And one of the things we realized very early on is that in order to be able to have the best offerings, we need to work very closely with clients and collaborators in order to tailor the solutions for their particular needs. And this is very much the thinking up to date, and something that we really have important in terms of how we approach the different workflows and our working relationships with the community. And so Our proteomics offerings, as I mentioned, initially we weren't really developing technology for proteomics workflows. So it's the first methods and products really were custom requests. So we developed prototypes that were then tested together with our collaborators at the time in order to make sure that they fit for purpose and that they have the performance and the characteristics in terms of throughput, scalability, and ability to automate. And really through their work, subsequent work in mostly in the cancer space, it gave us the credibility, it gave us the confidence and the platform to kind of build our proteomics portfolio from there. And over the years, we saw the expansion of these workflows to cover a wide range of organisms, sample sources, different tissues, cells, and so on. We saw the expansion of the workflows to cover different types of PTMs, as well as different types of affinity-based enrichment coupled to mass spectrometry-based workflows. We further highlighted the applicability of the workflows across different types of robotic and liquid handling stations. And again, through the inputs of the community and their lead, further these workflows were, there was added functionality added to these workflows in terms of the ability to automate them. We are constantly being involved in large-scale studies where we try to get a feel for what the community is requesting in terms of best practice methods, as well as how to process the data, as well as how to interpret the results. So we can go back, we can revisit our workflows, we can improve the performance, the throughput, and coverage. And DIA for us was a major game changer, similar to many labs in the proteomics world. Mostly because there was a large bottleneck largely removed at the LCMS stage, where previously we had analysis of between 60 and in excess of 180 minutes. Now with DIA, we see runtimes down to 5 minutes with very deep proteome coverage. So that's basically firmly puts the spotlight back on sample prep. So having high throughput and efficient sample prep was no longer a luxury, but an absolute necessity. And what's really important to highlight here again is that our ability to transform, adapt, and optimize our workflows to make them suitable for this higher throughput DIA analysis was really enabled by our close collaboration with our research partners. Their valuable inputs in terms of how we should develop the workflows. And so now we are seeing these workflows applied in the clinical space. And because of the ability to analyze large number of biopsies, biofluids in a robust manner, and we hope that the combination of high throughput sample processing with efficient LCMS finally unlocks the full potential of proteomics when it comes to clinical applications. Finally, a very fastly developing and hot area at the moment is the need to go deeper in the proteome, as well as go to lower input materials. So the so-called single cell or few cell proteomics applications, which is really enabled by the latest developments in mass spectrometry, having these more sensitive mass spectrometers. And again, through close collaboration with our research partners and collaborators, we are currently adapting and optimizing this workflow so we can have them applicable for these lower input amounts of material. And so we hope in the coming months, we will see more of this and we can make these available to the wider scientific community. And of course, these will be underpinned by the characteristics of robustness and throughput, either in manual or automated formats. And so with the slides, I think the main message I want to come across is that none of this would have been possible without the contribution of our clients, research partners, collaborators, as well as the wider omics and scientific community. So really, it was their ideas, innovative solutions, their feedback that is allowing us to put down these workflows and offer them to the community. And so this is essentially where the idea of the Emerge webinar series came about. We wanted to use now our networks that we built over the years to essentially provide a platform for emerging researchers to come and talk about their innovative solutions, their ideas, to come and chat to their peers about their research, and hopefully initiate some discussion around this and potentially some collaboration across different like-minded research groups. And so this platform is open not just to our current clients and collaborators, it's open to everyone. So if you feel like you want to come and talk about your research, talk about your innovative solutions in omics space, please get hold of me, and we'll be more than happy to have you part of this series in the future. And so for now, to the main part of the session, I would like to introduce our speaker for the day, Dr. Magnus Jakobsson from the Department of Human Technology, Lund University. So Magnus has a wealth of experience in protein methylation, going back to his PhD days, which he completed in 2013. He was focusing on protein methyl transferases, and then subsequently further honed his skills at the University of Copenhagen when he was completing a postdoctoral research fellowship under the supervision of Professor Jesper Olsen. And currently, Magnus is a co-leader as well as a group leader of the proteophones platform, as well as the group within the immunotechnology department of Lund University. And so today he's going to tell us about his focus in protein methylation and how he's applying these tools he's developing to solve both basic as well as translational research questions. And so Magnus, thank you very much for joining us today, and I really look forward to your presentation.
Okay, so thanks, Stoyan, for that introduction, and I'm really excited to be invited also as the first speaker of this eMERGE seminar series. And as Stoyan said, I will be talking about protein methylation and the new emerging field of protein methylomics. And to put this research in a context, I always like to start out with the central dogma of biology. So this states that the flow of information goes from DNA in our genes through a messenger RNA to a protein. And there's an increasing complexity associated with this process. But in turn, the proteins can be subject to post-translational modifications and engage in more complex higher order structures, such as in multi-protein interactions, and they can also be regulated in both space and time in cells. And it's this functional organization of the proteome that determines phenotypes at the cell and organism level. And it's often these phenotypes that we are interested in comprehending. And a key to doing so is then to understand these functional dimensions of the proteome and how PTMs regulate them. So typically PTMs are dynamic, and they are introduced by writer enzymes and can be removed by eraser enzymes. And a modified protein can in turn be recognized by a reader domain or a reader protein that mediate different functional outputs. And these readers can then modulate the activity, localization, stability, and interactions of a modified protein. And most PTMs that have some kind of regulatory potential have this complete set of writer, reader, and eraser proteins. So in the case of methylation as a modification, the writers are methyltransferases, the erasers are demethylases, and reader domains can, for instance, be Tudor and PhD domains. And also, for instance, protein acetylation and phosphorylation also have this complete set of enzymes that regulate the modifications and read out and mediate functional readouts of modified protein species. So going a bit deeper into methylation, so this is a representation of the human methyltransferase ohm, And the human genome encodes a bit over 200 enzymes with predicted methyltransferase activity. And these enzymes are often grouped based on their predicted structures. So the most famous group is probably this set domain containing enzyme group. So these set domain enzymes, they are mostly lysine specific methyltransferases that function in chromatin biology. So they methylate these flexible tails in histone proteins to and act as determinants of chromatin compaction and gene activity. But the biggest class is made up by the so-called 7 beta strand methyltransferases. And I will also showcase an enzyme from this class later in the presentation. And these 7 beta strand transferases, they are characterized by this topology. So they have 7 beta strands with alternating alpha helices. And most of these beta strands are parallel except for the 2 C-terminal ones. And all of these methyltransferases, they use the same methyl donor, the same molecule to donate methyl groups, which is called S-adenosylmethionine or just ADOMET. And this molecule is synthesized from methionine and ATP. So it has this methionine moiety to the left and an adenosyl moiety to the right. And that leaves a positive charge on this sulfur, which is a sulfonium ion, and that polarizes this proximal methyl group and makes it susceptible for nucleophilic attacks in nucleophilic substitution reactions. And these methyltransferases, they act by providing a scaffold that aligns nucleophilic substrates with this ADOMET molecule in a conformation that allows these nucleophilic substitutions to occur. And then generate a methylated substrate and a demethylated methyl donor. And there are many different flavors of methylation in proteins. So one of the most studied is methylation of the amino acid lysine. And here the amino group in the side chain, it can accept 1, 2, or 3 methyl groups, yielding mono, di, and trimethylysine. And these methylations are also reversible. So you have these demethylating enzymes that can remove them. Another key methylation event is methylation of the N-terminus of proteins. So the N-terminus of proteins also represents a primary amino group. So the chemistry is very similar to the side chain of lysine. And this N-terminal amino group can also accept 1, 2, or 3 methyl groups. But whether this modification is reversible or not has not yet been studied in detail. Then we also have histidine, and there are 2 nitrogens in this imidazole ring in the histidine side chain that can both be methylated. And finally, there is arginine, and for arginine, there are 3 different flavors of lysine of methylation. And we have been studying these methylations quite broadly, and here's a summary of the papers I have been involved in as a PhD postdoc and PI. So most of our work concerns lysine methylation, but more recently, I have worked with methylation of the protein N-terminus and methylation of histidine. And these modifications are a bit more exotic and emerging, and I thought that they were a good topic for this Emerge seminar series. So in this first example, this first example, it concerns methylation of a protein, a eukaryotic elongation factor called eEF1A. And this eEF1A protein, it performs a very central and important cellular function. So it brings charged tRNAs, so amino acid tRNA complexes to the ribosome to support translation and protein synthesis. And actually every amino acid that has been incorporated into every protein in every organism in life as we know it has been mediated by this elongation factor. So it really is a very important protein. And with that backdrop, it's easy to imagine that there's a tremendous evolutionary benefit to optimizing the function of this elongation factor through post-translational modification or editing. This elongation factor protein, it's reported to be methylated on several lysine residues as well as on the N-terminus. And very recently, we and other groups have identified several of the lysine-specific transferases that specifically modify these residues in the elongation factor. But I was also interested in identifying the enzyme that introduces this N-terminal trimethylation event, which is a bit more exotic. And usually the N-terminus of proteins are subject to co-translational acetylation. So N-terminal methylation is really an odd post-translational modification that I found particularly interesting. But to identify this methyltransferase that introduces this methylation in cells, I reasoned that the transferase would bind tighter to its substrate than its product, and that is that it would bind tighter to the unmethylated form of the elongation factor N-terminus than the trimethylated substrate. So I made some synthetic peptides that corresponded to either the modified or unmodified form and conjugated these with biotin and immobilized them on beads. And then did the enrichment analysis of soluble proteins from cell extracts and quantified proteins that were enriched by these different peptides using a label-free mass spectrometry approach. And this volcano plot shows proteins enriched by the unmethylated peptide on the right side and the trimethylated peptide on the left side. I found that a single methyltransferase protein of the 200 encoded in our genome was enriched by the unmethylated elongation factor N-terminus. And this protein was called methyl-13 or methyltransferase-like protein 13, and it was uncharacterized at this point. So to explore whether this methyl-13 catalyzes elongation factor N-terminal methylation in cells, I obtained a knockout cell model for the enzyme in HAP1 cells, and then quantified the degree of methylation at the site in wild type compared to knockout cells. And here are extracted ion chromatograms of a peptide that covers the N-terminus of the elongation factor 1A. And clearly you here see that the site is exclusively trimethylated in wild type cells, whereas the modification is absent in the methyl-13 knockout cells. So this demonstrates two things. So first, that the modification, it really does occur in vivo, and it's mainly trimethylated. And more importantly, it shows that the modification is dependent and exclusively dependent on the methyl-13 enzyme in cells. But to also provide a direct link between the enzyme and the substrate, I cloned the different domains of methyl-13. And for one domain, which I here denote methyl-13C, I observed that this domain was capable of methylating the elongation factor substrate in vitro assays. So here I incubated methyl-13C with the elongation factor substrate at different molecular, at different stoichiometries, and observed a dose-dependent methylation of the substrate. And you see here that it brings unmethylated substrate into trimethylated product in a dose-dependent manner. So collectively, this demonstrates or uncovers methyl-13 as the elongation factor 1A methyltransferase, and the activity is demonstrated both in human cells and in a more simple in vitro setting. And we were also very happy that these results were highlighted in a research highlight in Nature Chemical biology that goes on methyltransferase mediated regulation of translation. All right, so then I'll skip to the next part of the talk, which concerns histidine and the methylation of histidine. So here you see the structure of histidine. And the side chain, it has this characteristic imidazole ring. The imidazole ring has two nitrogens that are called N1 or N3. And both of these nitrogens, they can be both protonated and methylated in cells. And the methylated forms are called 1-methylhistidine if the methyl group sits on this N1 atom, or 3-methylhistidine if the methyl group sits on this N3 atom. And this is a modification that was first discovered in the late 60s, so it has been known for over 5 decades, but nothing has really been known about its enzymatic regulation and the extent and abundance of the modification. But very recently, or in 2018, the first human histidine methyltransferase was uncovered, and this was work from the Droszak lab in Poland and the Gosani lab in Stanford. And actually this year we have contributed to uncovering the second and third histidine specific human enzymes as methyl 9 and methyl 18. And these were published in Nature Communications and Nucleic Acid Research earlier this year. But aside from these 4 very kind of enzyme and single modification centric studies, very little is known about the abundance and extent of these histidine methylations on a system and organism level. And histidine methylation is particularly challenging to analyze, since there are no available affinity agents to enrich modified peptides for mass spectrometry analysis. So we instead devised an approach where we queried the most comprehensive proteomic data sets available for different types of methylation. And this allowed us to generate the database of histidine methylated sites in cells that we could then use to explore some fundamental characteristics of the PTM. And here we looked into the context and the function, the abundance, and also how histidine methylation interacts and crosstalks with other key cellular PTMs. And I will now show you a couple of results from this study, which was published last week in a new journal, a new sister journal to Nucleic Acid Research called NAR Genomics and Bioinformatics. So first I will show you some analysis we did specific for HeLa cells. So here we used the proteomic data sets that were generated by Dr. Becker Jensen in Jesper Olsen's lab, published in 2017 in Cell Systems. So these data sets correspond to HeLa cells treated with the different proteases, and proteolytic peptides were then subject to extensive offline fractionation before analyzed using short gradients and fast scanning MS methods. And we then requeried these data sets for the different flavors of methylation, which in this case is methylhistidine and methyl-lysine arginine, dimethyl-lysine arginine, and trimethyl-lysine. And in HeLa cells alone, we uncovered a bit over 2,000 methylation events. So we found almost 300 histidine methylation events, almost 900 lysine methylation events, and slightly more arginine methylation events. And since there are no available affinity agents to enrich lysine and histidine methylation, these numbers actually represent the most comprehensive resource for these modifications generated to date. We then benchmarked our identified sites to the most comprehensive databases available online. And for PTMs, this database corresponds to Phosphosite Plus. And most of our, or essentially all of our histidine methylation events were novel, and most of our lysine methylation events were novel, whereas about half of our identified arginine methylation events were novel. And we then explored whether these modifications were kind of concentrated or frequently occurring or on single proteins, or if they were scattered across the proteome. And the results are illustrated here. So here we show the number of methylation events identified for protein for the different types of methylation. And clearly most proteins contained a single methylation event, suggesting that they are indeed scattered across the proteome. We then looked into the abundance of methyl proteins, and here we found that all flavors of methylation occurred on proteins that were more abundant than the average protein. And this could have two alternative explanations. So first of all, I mean, acquisition of single peptides or analysis of single peptides by mass spectrometry is related to an abundance bias. But more interestingly, several methylated proteins are highly abundant and regulate key cellular processes. And many abundant or many methylation targets in cells correspond to, for instance, elongation factors, histone proteins, key chaperones, ribosomal proteins. And it has also been hypothesized that these abundant proteins, that the function of these abundant proteins is fine-tuned by these methylations. Then finally, we explored the subcellular localization of the proteins that carried the different types of methylation. And here we observed that both histidine, lysine, and arginine methylation were distributed throughout or across the different subcellular structures, and also at comparable frequencies. And then we looked into which domains and sites that were targeted by methylation, and domains that were significantly enriched for a methylation event are indicated in this Sankey plot. So here you show domains, and here you see domains for histidine methylation, arginine, and lysine methylation. And I first wanted to highlight that some of the kind of expected culprits came out. So lysine methylation was found to be overrepresented in histone proteins, and it's also a well-characterized epigenetic mark. Arginine methylation was found to be most significantly over-enriched in RNA recognition motif domains, and this is also where it's been suggested to play a key function in the literature. And histidine methylation was a bit more scattered, but one striking feature was that several of the domains that were enriched for histidine methylation turned out to be zinc binding. And also looking into the primary sequence context of these histidine methylation events, we found that they were present in a primary sequence context of repetitive histidine residues. So with histidines in -2, -4, +2, and +4 positions. And this is actually exactly the motif that is responsible for coordinating zinc in these zinc binding domains. And this motif, it also matches well with the literature, and in a recent paper in Nature Communications, it was reported that this new histidine methyltransferase methyl-9, it catalyzes methylation, but it requires this histidine X histidine motif where X is preferably a small amino acid. And our identified motif matches perfectly to the motif for methyl-9 with a serine spacing the two histidine residues. And then Similar analysis for arginine methylation and lysine methylation also revealed interesting features. So arginine methylation was found to be enriched in glycine and protein-rich domains, which has also been reported previously in the literature. Lysine methylation, on the other hand, was found to be enriched in acidic residues, so with a high frequency of aspartate and glutamate up and downstream of modified sites. All right, so now I've presented results from our protein methylomics study on histidine methylation and talked about the context, the function, and the abundance of these methylations. And now I will just show some of our analysis concerning PTM crosstalk and the interaction. And we explored crosstalk between histidine methylation and phosphorylation. And to that end, we used a great resource that was published by Ochoa in Nature Biotech last year. And what they did in this study was that they reanalyzed over 7,000 experiments, phosphoproteomic experiments deposited in the PRIDE database. And they identified over or almost 120,000 human phosphosites. But then they applied a sophisticated machine learning based approach to assign a functional score to each of these phosphosites. And they did that by looking into the regulation of the sites, their conservation, evidence, and where they are located in structures. And the output was a functional score attributed to each of these sites that reflect the functional importance of the phosphorylation. So we filtered out phosphorylation sites from this resource that co-localize with our methylation events. So here are density plots for all of these 116,000 phosphosites and the ones that co-localize with histidine, lysine, and arginine methylation. And you here see that the methylation sites that co-localize with a phosphorylation, they all have an above average functional score. And to highlight one interesting site we found, I chose this one. So it's a histidine methylation site in position 75 of the actin protein ACT-A1. And it then has this high scoring functional phosphosite in tyrosine in position, in an adjacent tyrosine in position 71, which has a functional score of 0.62 in the analysis. But this the histidine methylation site is located in a loop in actin. So actin is an ATP dependent protein, and its polymerization is dependent on ATP hydrolysis. And this loop that harbors the methylated histidine, it has been reported to have a function in sensing ATP and ADP binding in actin. So it's a key and central features related to actin activity. I then looked into PTM databases and also databases with disease-associated SNPs to see what else went on in this loop. And we actually found a range of different PTMs in and adjacent to this loop. So the different acetylations, ubiquitinations, different flavors of methylation and phosphorylation, as well as disease-associated polymorphisms. And most of these modifications were also present and reported to occur in mouse. So this would then support the model where I mean, single or multiple modifications in this loop in actin collectively may regulate the function of actin. So this could represent an actin code in analogy to the histone code in epigenetics. And this combinatorial action and interplay between PTMs and particularly histidine methylation is something we find very intriguing and are excited to follow up in future studies. All right, so finally, I would like to show you just a single slide on what we are doing on lysine methylation. And for lysine methylation, we are applying more conventional approach for studies, which is based on affinity enrichment of modified peptides using immobilized affinity binders, and then analyzing enriched affinity or analyzing enriched material by mass spectrometry and informatics. But the generation of binders for lysine methylation has been a challenge for the research community, and many labs have tried, and still there are no available robust binders for this PTM on the market. But we have used a new immunization approach and actually been able to generate binders that specifically bind to and recognize either monomethylated lysine, dimethylated lysine, or trimethylated lysine. And we are now working on functionalizing magnetic beads from resin bioscience with these different antibodies to enrich lysine methylated peptides in a robust and reproducible and automated way for proteomic studies. And this is something we are also really excited about developing moving forward. Okay, and finally, I would like to acknowledge my research team. So Julia, who did some of the work on histidine methylation, and Albin, who has been working on lysine methylation. We are actually recruiting a bioinformatician. So if you are out there or know anyone, please reach out to me. As Stoyan also said, I co-lead a mass spectrometry platform called Proteoforms at LU. And our focus is analysis of PTMs and protein variants. So if you have questions or queries related to those topics, please contact us. And finally, I would like to thank Resin Bioscience for the invitation to present here today. And I would also like to highlight Sebastian Kapell at Karolinska in Stockholm, who did most of the PTM informatics I presented on protein methylation. And with that, I would also like to thank you all for listening and for your attention, and I would be happy to take any questions.
Fantastic. Thank you very much, Magnus. I think it's a super interesting presentation for myself as well. So maybe I can kick things off with a couple of questions. So it seems like one of the challenges with studying methylation is obviously how to enrich for these proteins or peptides, because there's no obvious handle there. You've got, it's not like phosphorylation where you've got a charge to pull on. So do you see a one preference of a method or methodology to cover the different types of methylation sites? It seems like you have a preferred approach for type of methylation, let's say methylation for lysine versus histidine, or is there something more universal that could be applied? And also maybe a follow-up question in the data set that was the HeLa methylome. I noticed that there was different enzymes. Was there preference in terms of how many methyl sites were identified based on the enzyme? Because I suspect with trypsin you'll get more miscleavages because of the if it's on lysine or arginine methylation. So maybe some of the other enzymes are better suited for this type of application.
Yes, that is great question, Stoyan. So I will start with the second one concerning enzymes and proteolysis. So yeah, many PTMs they block proteolytic cleavage with the commonly used enzymes, and trypsin is inhibited by both methylation and acetylation of lysine, for instance. So that is correct. You would expect one missed cleavage after a methylation event. But that said, I mean, trypsin and lysine are also the most efficient enzymes for proteolysis in MS sample prep. I mean, at large, different enzymes, they give complementary results and will give you coverage of different sites.
Okay.
And then for the first question regarding different types of affinity agents one can use for methylation. So there has been single studies from single labs that have used, for instance, recombinant methyl-lysine binding domains for enrichment. And there are also labs that have generated batches of polyclonal IgGs that seem to have worked out fine in single applications. But there seems to be stability with these recombinant methyl-lysine binding domains over time. So a preferred approach would be to have a recombinant affinity agent, like a recombinant antibody, where you can always go back to your E. coli stock and produce fresh antibodies. So that would be one approach. And then, I mean, these molecularly imprinted polymers also seem to work well to enrich for histidine phosphorylation and tyrosine phosphorylation. So that would be another interesting approach to evaluate for these methylations.
Yeah, no, absolutely. I think those are quite interesting to try as well. And so we have another question from Emma. Is asking, how do your lysine methylation affinity reagents compare to the others currently available, like cell signaling or PTM biolabs? I suppose have you done some benchmarking against those?
Yes. So I mean, we have tried. We have also cloned so there are particularly two methyl-lysine binding domains that have been implicated in the literature as candidates to enrich modified peptides for mass spec. So that is one domain called 3xMBT. That's a dual affinity domain that should recognize mono and dimethyl-lysine. And then there's another double tutor domain of a Jumonji D2A protein that supposedly recognizes trimethylates in a context-independent manner. So what we have cloned these domains and optimized expression boundaries and expression systems, but in our hands they have not been useful in enrichment.
Okay, fantastic. Yeah, so we don't have any more questions for now. If anybody comes up with more, you can contact me or Magnus. I see another one here. What is the functional relevance of N-terminal methylation of eEF1? And I lost it. I lost the question somewhere. They're in the chat. Sorry, there we go. What is the functional relevance of N-terminal methylation on eEF1 A1 in elongation processes?
Yes, so biochemically the methylations they increase the void occupancy and the hydrophobicity of a modified lysine or a modified N-terminus. And the N-terminus of proteins, it has a pKa value around physiological pH, and trimethylation stabilizes a charge on the N-terminus, so it generates a constitutive positive charge. So that is a biochemical answer to the question. But functionally, we observed differences in translation dynamics in metal 13 deficient cells compared to wild type cells. So we performed ribosome profiling experiments or ribosome footprinting experiments, and then looked at the occupancy of different mRNA codons in the acceptor site of the ribosome. And we could then assess the speed of translation for each codon on a global level. And for selected codons, we observed a decreased speed in translation.
Fantastic. Okay. If there are any more questions, we have some more time. Otherwise, we can handle any questions afterwards. I'm sure Magnus will be able to answer those. And as I mentioned at the beginning of the presentation, we will make the session available so you can view it at your own time. So okay. in this case, there's no more questions. I want to thank you all for coming through for this first episode, hopefully first of many. And of course, I want to thank Magnus for agreeing to be the first person in this new series. It's really appreciated. And then hopefully we can hear from you more in the future as well. So yeah, I'm going to end it here. Thank you very much, everyone.